Welcome to the second part of Chapter 20. So we're going to focus on some of the genomic aspects where we analyze and try to process the genes that are part of the entire DNA that makes up an organism or a species. And so as we've gone through this process of gene cloning, where we've isolated genes of interest, we want to store these for later use. We don't want this to be a one-time deal. So we'll oftentimes put these in bacteria, artificial chromosomes. So these will just be really large plasmids. So there's large circular pieces of DNA. And we're typically going to do these, not always, but typically, especially in eukaryotes, we're not just going to take the gene and throw it in there. We're going to take the mRNA transcript that's been produced, that's been processed, that's had the introns removed, and we're going to take and reverse transcribe that. So we're going to convert that mRNA to what we call complementary DNA. So it's DNA, but it's just made from an mRNA transcript, so we know it's minus those introns. We can then insert this in that bacterial artificial chromosome and know that even if we gave that new plasmid, even if we gave this to a prokaryote, it wouldn't have difficulty because the introns have been removed. So you'll see coming up we'll talk a lot more about cDNA just because it bypasses this issue where a prokaryote would not be able to process a eukaryotic gene without this additional step. And so we normally just go ahead and take the mRNA as it's already been processed, convert it back to DNA to eliminate that issue. Now, for DNA analysis, we've got several different types. This will just be a quick intro, then I'll go more in depth. So we'll have PCR, polymerase chain reaction. This is to basically make a bunch of copies. Gel electrophoresis is going to separate out fragments. So when we cut stuff up, we can ultimately look at those fragments by separating them to see which ones are present. RFLP analysis, this is going to be restriction fragment length polymorphism. So this is going to be another way of ultimately cutting the stuff up, which we can then analyze using something like gel electrophoresis. So this is mixing restriction enzymes in with DNA. And we've got southern blotting or other blotting where we take the data from gel electrophoresis and the RFLPs that we've kind of processed on this gel and we transfer it onto a paper or take a picture of it so we now have it like preserved. You know, we now have something that we can store that's not actually a hunk of gel. So a little bit more in depth here for the other ones then. We have genomics. All right, this is just a real quick intro. I'm not going to go into the in-depth things of how we do it. But for genomics, this does entail, in most cases, sequencing the entire genome of something at some point. This is kind of our goal. We've done this for humans and many other animals that we normally use for study. So lab mice, fruit flies, Arabidopsis for plants. It's a little mustard plant they use a lot. Many agricultural organisms we've gone through and sequenced their genome so we can actually look at the exact bases, the exact nucleotides that they have to try and locate things like genes, as well as to help try to figure out where else things differ, to try to figure out some even of these intronic differences, some of these other areas that we know don't code for genes, but we're not sure if they're regulatory, we're not sure if they're essentially not doing anything. We don't know what their purpose is exactly. So by learning more about what those sequences are, and perhaps some commonalities between organisms, we might be able to figure out what this other 98% does a little bit better within at least the human genome. In other genomes, it may not be exactly 2% coding. It could be more than that. But in general, there's a lot of DNA that doesn't appear to be coding for proteins. And we want to figure that out. So the Human Genome Project took, I believe it was 5 people and sequenced their genome and then compiled it so we have a human genome. And so they've been using this to try to figure out where genes are located and then eventually, hopefully, figure out essentially what each gene is capable of doing. And so that way we can target things better for stuff like disease. Now, coming up after this, we've got the idea of the technologies that they use, both for the testing we've been talking about, even sequencing in some cases, where we're going to process this information. So PCR, we're going to ultimately go outside of a cell and make a bunch of copies of some DNA piece. So the general rules, heat it up. By heating it up, you split it, so these hydrogen bonds rupture, so we now have 2 single-stranded pieces. You then allow for it to cool a little bit and mix in some heat-tolerant DNA polymerase, some nucleotides, and ultimately some single-stranded little pieces of DNA that will act as primers. And so once you've done that, You can then allow the polymerase to do its job to make copies, and so now we have 2 copies of the DNA that we're after. Sometimes you'll do this for a whole bunch of DNA, sometimes you'll do it just for a gene. You can target it based upon things like the primers. And so once we do this, we can then go through the process again. So we can then heat it up, make sure we add in the polymerase after we cool it down a little bit, make sure there's plenty of nucleotides present and those primers, and it'll go through and copy itself again. So we can do this over and over, and we can mechanize this where every so many minutes this process will finish and do another round. And so each time it doubles the amount of DNA that we got before. And so this allows for us to take a very small amount of DNA from something like fossils, where there might be just a little bit of blood left in some bone tissue, forensic, like the hair on the hairbrush, or in some cases diagnosis, especially prenatal diagnosis, where we only have like one cell we can pull off of this embryo to try and run tests. So with this, we can take that small amount of DNA and really blow it up into a much larger amount of DNA, which is useful enough to run multiple tests. Now, gel electrophoresis is going to be where we're going to take, and this is kind of tied with RFLPs, but we're going to mix in a bunch of restriction enzymes, specific ones. We're going to cut up the DNA. We're then going to put it in this little well, and we're going to put it next to the negative, well, that'd be the anode here, the negative terminal. And so DNA is slightly negative, and so it's going to be pushed away from the negative and towards the positive. So it's going to try to move through the gel. Now, the bigger the piece that you have, at least for DNA typically, the slower it's going to move. So when we're done, we can stain this, or we can use like a fluorescent dye so it'll fluoresce with UV light. So we get this banding pattern based upon the fragments. So the bigger fragments show up as a band that's closer towards the start, and the smaller pieces show up as bands further along. So this one's showing very few, but when we get to the DNA fingerprint, you'll see there'll be a lot more bands that you'll be able to pick up. And so we can use this to kind of figure out patterns and figure out what fragments are there for analysis later on. So that's gel electrophoresis. This is just the device. This is, you can see it's hooked up electrically, and then that's the gel. In this case, they just put ink in there, and it just kind of separated out the different inks that were present. So that one's not by any means crazy elaborate, but it does kind of show you what it's doing, and you can see that the different pigments do have different sizes or different charges. I mean, if you have a much different charge, you can get more of a push. But something is certainly different about these, that the greens and the yellows manage to get so much further than the reds and the blues. So RFLP, this is where we're going to take a bunch of restriction enzymes, as I said, specific ones, and cut up the DNA into fragments. We then, by running it through a gel typically, are going to be able to analyze which fragments are there by doing a blot. And so a blot's going to be either, I believe it's called nitrocellulose paper is the common paper that they can put on the gel to transfer the stuff onto the sheet. You can also just have them fluoresce, so you can put them under a UV light if you have fluorescent markers and take a picture. Either way, we now have, hey, I was right, nitrocellulose. But either way, we have the ability now to manipulate it because we have it on paper, if you will, instead of just me hauling around a chunk of gel, which doesn't usually show as clearly these markers. And so we'll call this banding pattern that we see here a DNA fingerprint. And we'll oftentimes run in one of the columns known markers, and then in the other ones we can run, you know, the person perhaps that we're testing it for, and then the blood from a crime scene, or we can run like the mother stuff in this column, we can run the father stuff here, and the babies in the third, so we can try to make sure that if these two are the parents, the banding patterns, if we kind of add them together, should have all the same bands that the baby possesses. There might be some extras, that's okay, but there should be all the ones that the baby possess should be present. This is also how sometimes they can tell that the blood that they've collected from a crime scene isn't this person, but they might be able to figure out that it's a relative of that person because so many of the bands are in common that it appears as though that it's a close relative. And so you didn't do it, but they're going to be giving your brother or sister perhaps a knock at the door. And there are other types of blots, so we can do this whole process of cutting things up, running gel electrophoresis, and analyzing the aftermath, these banding patterns. We can do that for mRNA. We call that a northern blot instead of a southern blot. And we can do that for protein, which we would call a western blot. So just so you're aware, these technologies don't just apply to DNA. That's usually what I see them used with. But you can use them with the other guys that we've talked about, mRNA and proteins. Now for gene expression, I want to know not just what genes you have, but what genes you've turned on. So what we're going to do here is do reverse transcription RT-PCR. So we're going to take mRNA, convert it to cDNA, and then that way we can go through and cut up and analyze what cDNA pieces are present. You know, we can amplify them, we can do the same normal process to try to figure out which ones are actually turned on, which ones are being transcribed and therefore have the opportunity to be translated. And so that's really the only difference with an RT-PCR, is that you're not just focusing on do you have a gene or making a bunch of copies of a gene you know you have. We're now trying to go through and see, are you using a gene, and I want to make a bunch of copies of the guy that you're using so I can then try and test it to figure out exactly what gene that is. So this gives us some insight into things like epigenetics, where we're focused on is the gene on or off, not just do you have it. And so on a grand scale, we can do a DNA microarray assay. Big word, scary word. I'll try to lay this out smoothly. So the way this works is we're going to take and do essentially an RT-PCR. So we're going to take the mRNA, usually from two different cells. So for instance, you have some type of disease cell like cancer cells, and then normal cells. And what we're trying to do is figure out how are they different so we can target those differences, so we can figure out what went wrong. So we're going to analyze and collect the mRNA. So that's what we've talked about before with the whole RT-PCR. We'll make copies of it as needed and convert it to cDNA. The difference is in each sample we're going to give it a marker, so we're essentially going to use special nucleotides that have been altered in some way or had a marker stuck to them, so that way we can detect them. And so in this case you'll see it's common to use red and green. And so the cancer ones will show up with red, and the normal cell ones that are turned on will show up with green. We then take and use what's called a biochip, which is just a chip that's been essentially printed, but it's been printed with nucleotide sequences on it. So these single-stranded nucleotide sequences where each section has like its own unique sequence. So there's literally regions on this with their own sequence, and those correspond to genes. So what we're going to do is take this stuff, pour essentially this result from our RT-PCR, so we have a bunch of copies of each of these guys, these cDNAs. We're typically going to want them to be relatively single-stranded here, be single-stranded, and we're going to pour them onto these single-stranded DNA pieces that are on the biochip. Now, if they bump together with someone that's the complement to them, so if they find a sequence that corresponds, so it's essentially saying that we've turned this gene on, so I have a sequence from this gene that I've gotten up here, right, from my mRNA, that's now cDNA. So if I can find the complement, if that gene is on, it'll stick to that gene sequence on the biochip, which I should know this is that gene sequence, so I can tell which gene it is. We then kind of rinse this off, just to make sure that nobody that didn't stick sticks around. So what's left now is a bunch of these cDNA pieces, these single-stranded pieces, that have gripped hold of a complement that's on the biochip. I then take lasers and shine lasers to see who lights up. And so if you have a gene that's only turned on in the cancer cells, it'll light up red. So you'll see like a red dot on the biochip when the laser scans it. If I've got one that has only been affected by, or only stuck to, it's only on the normal cells, it'll show up as green, which I don't have a really easy way of doing. I guess we'll use blue for now, so cope with it. That's green. And if it's been turned on in both of them, it would show up essentially as a yellow. If you mix red and green, you get yellow when you're talking about light. And if none of them have it on, it just shows up as black. So we can then essentially look at the results from this laser scanning the biochip to see where it's black, to see where it's red, to see where it's green, to see where it's yellow, to see what the differences are. If they're black, where neither of them express it, that tells you it's not a difference. If they're yellow, where they both express it, no difference. But if you see it red or green, that's where we kind of throw up the flags and say, all right, we need to focus on those and try to figure out why they're different, and perhaps that's where the clue is that will let us figure out why that person has cancer, what went wrong that's led to this. And so we will continue to study these expression effects, and this is kind of cool because when we do this, it's somewhat close to torture, but it's interesting still. And so what we'll do is we'll do in vitro mutagenesis, so we try to mutate genes while it's still like developing, so as it grows we can see what happened. We can be like, all right, let's mess with this gene and see what the effect is. So that can help us figure out what a gene does. And so if we can manage to turn it off or alter the gene or literally just mess it up so much it doesn't produce the protein, we can start to see what the gene did. The other way is if we know the exact sequence of the gene, we can try to build a molecule that will go after the mRNA it produces and destroy it. So we call this RNA interference. And so by doing this, by destroying the mRNA consistently, we can make sure that we don't allow any translation to occur. And so once again, you still have no expression, and we can see what the damage is. We can see, did it affect it? If it did affect it, what part of it was affected? So these are some of the ways that we can take genes that we know exist in the genome and try to figure out what they do within the genome. Now this is going to be done with things like fruit flies and mice. This is not typically going to be done with people for obvious reasons that you can't just mess up people to see what happens. That's why we focus so much on things like the microarray assays, where we can study people that already have a problem versus inducing a problem in them, which is not ethically sane or okay. So the whole reason why we get into this process and we deal with all this stuff and we screw around with all this is there are some huge benefits that we can get from our technology. And so in this case, we can have diagnosis. So if we know genes, we can potentially develop tests so people can figure out if they have genetic disorders or predispositions. So we could either treat them eventually if gene therapy works, where we kind of use viruses to infect people with a healthy gene. So eventually, if we get there, if we can diagnose it, through gene therapy, we might be able to fix it. We've got vaccines that we're working on to try to make sure we can get rid of diseases just in general. You've got forensic, like crime stuff that they're doing, or even anthropological stuff, where they're trying to figure out like the genetics of groups of people that have lived a while back, if they can get DNA, so they can try and trace kind of how humanity has spread and evolved. We've got transgenics, which is both genetic engineering. Transgenic you see more referring to animals. Genetic engineering, they normally refer to that a little bit more in terms of plants. But both of these are trying to improve, especially agricultural stuff, to keep us nutritious, to keep us fed. And then lastly, we've got stem cells, trying to figure out how we can affect gene expression and how we can trigger cells to become stem cells. Because if we can master stem cells, these guys that can be anything, there's so many disorders, there's so many accidents, there's so many issues that we could maybe fix. I can't tell you for sure we can fix all of these, but certainly there's a good chance that we can at least fix some of these, and some of these disorders have very little current hope of being fixed elsewise. So this is one of the big fields that holds a lot of promise for a lot of disorders anytime in the near future. And so there's a lot of discussion about this, and some of the work is actually trying to avoid that discussion. And what I mean by that is they're trying to do things like convert a skin cell from you into a stem cell, or collect stem cells from you so they don't have to worry about people talking about embryological stem cells or umbilical stem cells or anything to do with babies, where a lot of people start to have ethical concerns. And so if I can just go through and get the stem cells from your fat, most people aren't going to whine about getting some liposuction so they get stem cells and regrow you an organ. Most people are going to say that's a win-win. Hope you guys have enjoyed 20, and I'll see you guys soon.